really have no announcements. And I will turn it over to David Wake to introduce today's speaker, and then Jim McGuire will moderate questions at the end. I'll just remind everybody to stay on mute throughout the seminar so we don't have any feedback or background noise. Okay, David, it's all yours.
All right, thank you. It is a pleasure to introduce David Blackburn, Curator of Herpetology at the Florida Museum of Natural History in Gainesville. David is a leading amphibian biologist, known especially for his many important papers on African frogs. A graduate of the University of Chicago, David started out under the mentorship of my former student, Eric Lombard. I narrowly missed being his graduate sponsor because he applied to work with me. I stupidly and idealistically told him I was soon to retire, would not be taking new students. I did not realize that my most productive research decade lay ahead. Marvelly and I took our last sabbatical the next year. She is a Radcliffe Fellow and I as an Alexander Agassiz visiting professor at Harvard. And there I met David Blackburn, who had become a graduate student of another former student, Jim Hankin. Hankin had just been named director of the venerable Museum of Comparative Zoology, and he was preoccupied, to say the least. I became something of a surrogate mentor of David's and a few others, and we organized a discussion group. He also took a course that Hankin and I taught. And the friendship developed that continues to this day, I'm happy to say. David did a multi-year postdoc with Linda Trube at Kansas as part of the multi-campus, including Berkeley, Amphibian Tree of Life project, and he was highly productive. He accepted a curatorial position at the California Academy of Sciences in San Francisco in 2011, and he was with us in this area for a few years. He then left in 2015 to accept his current position in Florida. David has high impact on the field of amphibian biology with his excellent research program, his graduate education, and his many publications ranging from morphology and paleontology to molecular systematics, phylogenetics, and biogeography. He has been involved in a wide range of activities and organizations and currently heads the overt Thematic Collections Network, sponsored by NSF. This will be the topic of the seminar today, Digital Specimens and the Future of Natural History Collections. Welcome, David.
Thank you, Dave. I really appreciate that introduction. I am thankful that David Marvelly ended up in the office next to me when I started grad school. And the seminar that Dave mentioned with Jim was also my first introduction to a lot of other amphibian biologists because I came from a paleo background. And that included folks like Vance Breedenberg and Tyrone Hayes actually came through that semester as part of that seminar as guest speakers. All right, hopefully this doesn't all go wrong as I share my screen. Okay, share. You should see camels. Now you should see a fish. Can anybody give me a thumbs up that they see a fish? All right, thank you, Becca. Great. All right. So as Dave noted, Dave invited me to speak about a specific project. And so that's what I'm going to do. I'm going to sneak a small amount of related science in at the very end. All right. So like many of you, I spent a lot of time in the field making physical objects to put in collections for our research. Sometimes this even has a MVZ connection. So here's Becca Tarvin and former MVZ grad student of Jim's, Dan Portick, in the field in Cameroon. And, you know, like many of you, a goal of being in the field is creating these objects, these specimens that we use for our research. And now, you know, it's I guess a popular phrase to say they are extended specimens, right? So we have the physical objects, and we have photographs, and we have calls, and we have parasite data, and tissue samples, and all those things linked to physical specimens like these that are the products of our field research. And those physical objects go into these spaces and these collections, and, you know, like many of us oversee and manage. So, you know, in the past 20 years, the information about these specimens that are hard won in our fieldwork, that information is more accessible than it's ever been. Even when I started as a grad student, which wasn't that long ago, none of this really existed. I mean, I, at that time, used the collection, I can't even remember what it's called that Jens started. What is the Herpetological Collections database? That was just a list of, you know, which institutions had how many specimens of a species, and it was really incomplete. I mean, now we are a far, far way away from that. So the information that is there for our specimens is more accessible than ever, but the physical specimens themselves are about as accessible as they were in the 19th century, right? Like, they depend on us to visit them in person, or they depend on being put in the mail and sent to a person. And in a given year, if you had to loan one frog, you know, well, especially if you're someone like myself who doesn't return loans for a long period of time, you're very lucky to, you know, that specimen to see more than one researcher in a given year, right? Their ability to share a physical specimen is quite low. You know, so for those of us that are interested in phenotypes and at least anatomical traits in this case, you know, we're interested in taking these specimens and doing things with them, right? We want to actually see internal anatomy. We may want to dissect them. We may want to skeletonize them. We may want to clear and stain them. We may want to focus on a particular part of their body, such as in this case, the skull of this frog. We might take qualitative data, but we also might be interested in quantitative data for those same specimens, such as the thickness or density of those bones. And we might want to have those type of quantitative phenotypic data for analyses of evolution, right? In addition, we might want to see things that aren't really there. We might want to visualize spaces, in this case, the inner ear of this frog. See things that are really difficult to do unless you actually have the animal in hand. But many people aren't interested in the hard parts. They're interested in soft parts as well. They might be interested in the surface area, the volume of these animals, the skin itself, the texture of the skin, or the other parts of the organism, right? The soft parts. We now have the ability to digitally take these animals apart into different organ systems. We can separate them into cardiovascular system, muscles, nerves, put them back together again. And the way that we do this, at least in my lab, is mostly using CT scanning, although not exclusively, but predominantly CT scanning. So we take these specimens, and then using X-ray CT scanning, which I'll come back to in a minute, we can create these slice images that we can create all of these products that I just highlighted, from skeletons to spaces to quantitative data, even to 3D prints for outreach. Using other approaches where we can actually see the soft tissues of these animals, where we take these animals, we soak them in something like iodine that serves as a contrast agent to increase the density of those soft tissues. We can get all these other products, from skins to muscles, nerves, cardiovascular system, and even things like eggs or parasites inside of these animals. Sorry, I'm trying to figure out which slide I'm on. Okay, great. So just as a theme of a lot of the work that we've been doing as part of the project I'll talk about is creating what I kind of like to call these digital collection objects, right? These are really, they're digital surrogates. They're not exactly, right? There's a processing step, a step of imaging the animal that may lose some information from the animal as part of that process, but it's trying to be a faithful representation of those things. And being able to have those, not just a two-dimensional picture, but a three-dimensional volumetric data set. So it's not just the surface of the frog, but it's all of those internal pieces as well. These data really add value to these natural history collections, and that's going to be a theme towards the end of the talk. One of the things that I'm really interested in, also relates to my role in IDIC Bio, is increasing and democratizing access to collections. And I think one of the things that's been most fulfilling of the course of the overt project has been two things. One is seeing how many other disciplines that normally don't use our collections have started using our collections because they're using these digital objects. And the other part about that's really, to me, exciting is that we're also seeing a lot of non-researchers using these collections. So they're using these collections, especially for education at all levels, from K through 12 through graduate school and beyond. So just going to go over a few basics of CT scanning, just for those of you that aren't really familiar with this. So for most of the CT scanning that we do, and my impression talking with folks at Berkeley is that there's, I guess, in the works, a purchase of a CT scanner that would be sort of similar like this. This is a CT scanner that produces from a filament a spew of electrons that are focused through a magnetic lens and hitting a target, things like molybdenum. And out of that target, we have a spew, a cone beam of X-rays, which remember are photons being created. And those photons go across a space to a detector plate where those photons are converted back to electrons. If we put an object in the middle of that cone beam, like this specimen, it's effectively casting a shadow on that detector, and it's attenuating the X-rays. And so one of the things that we're seeing is we're basically seeing the difference of the X-rays that leave the detector, or sorry, leave the source and then get to the detector. And so it's that difference that's enabling us to see the shadow of the physical object. And if you, another point that's important here is that the way magnification works is if you wanted to make this specimen larger, is you actually would move it to the left, closer to the source, and it's actually going to magnify the view that you get on the detector. Likewise, if you wanted to make it smaller and have a decreased resolution, you would move it closer to the detector. In our project, this is something we use a lot to get just a head only, a high resolution skull scan, and then we move it farther away and we get a whole body scan. That's at a lower resolution, but it's closer to the detector plate in order to do that. In most CT scan machines that are used across many academic institutions like ours, we have an object that sits on a little stand, and that stand moves around in a circle. You'll see this smiley face is actually a sphere and is casting a shadow on this detector plate. And as the specimen rotates, the machine is capturing separate X-ray images, right? And so it's that set of two-dimensional X-ray images that are the basis for the data that we're going to be talking about. So here is an example. On the left, as you see this rotate, this is just simply a series of two-dimensional X-ray images, right? There's no volume here. There's nothing three-dimensional. Your eye is interpolating and actually following those pixels around in a circle and recognizing it as a three-dimensional thing. The computed tomography part of this is actually following those voxels of, representing different dense parts of the animal, following those and creating a three-dimensional volume that we can then reconstruct and get these actual three-dimensional objects out of. So the data from a CT scan is effectively this series of grayscale images that provides us a map of the density in a given volume. And it's out of that we create all of these three-dimensional models that we'll talk about. So a nice part here is that, once we have this volume, we can slice and dice it any way we want. We can dissect it in traditional anatomical planes like this, or we can view it in any other plane that we might want. And out of that volume, we can also begin to realize the most dense parts, the soft tissues and the hard parts of these animals. You can take a data set like this and open it up in a piece of software like this. This is volume graphics, but there's other pieces of software you've probably heard of, like a Viso and a Mira, Dragonfly, now the free and academic software, a Slicer, which is really great. And with some training, you can very quickly go from a data set right off the CT scanner to a three-dimensional object, to a thing that you could save and print on a 3D printer as, say, a mesh file. So this is Ed Stanley. I'm just going to recognize Ed here. Ed is critical to everything we do at Florida. He was a postdoc in my lab at the CalCAD and at Florida, and is now a faculty member here at Florida, running our digital imaging lab inside the museum. And training students to quickly do this work is a fundamental part of what Ed does here at the museum, including teaching courses on CT scanning. with those 3D data in hand, we can really open the doors to our collections. We can look at really rare animals. So here's a Sicilian from Marvelly. This is the only specimen of this species in the United States that I'm aware of. But yet I can put it in an email as a 3-dimensional thing and mail it to Marvelly. And I think that's really a phenomenal advance, right? And, you know, we only have one of them. We would never cut it up. We're not going to skeletonize it. We're not going to clear and stain it. We're probably not going to put it in the mail to loan it a lot of times, but yet I can email it. And I think that's a really powerful transformation. Another fun part of doing CT scans is finding things that are unexpected. Well, in our lab, we refer to as natural history bycatch. So here are 4 frogs that all have things in their stomachs, you know, and sometimes unexpected things. So on the top left, this is the first record of this family eating another vertebrate. There's a Phrynobatrachus eating an Arthroleptus, a Barbarula from Palawan in the Philippines. Barbarula is already known to eat crabs, and here you go. CT scan Barbarula. You often find whole crabs in their stomach. And on the bottom are 2 of the 2 different species and 2 different genera vying for smallest frog. And a lot of these animals we have 0 natural history for, or very little, especially when it comes to diet. So on the left, here's a Paedophryne from New Guinea that has a single ant in its stomach. And on the right is a Brachycephalus from Brazil that has a collembolin that's effectively the size of its brain. And, you know, a dietary study based on one thing is obviously not, you know, up to snuff with a lot of, you know, modern ecological studies. But for a lot of the taxa that we're interested in, we have literally 0 information about natural history. Sometimes those things come out of CT scanning. In soft tissue CT scans, we also get things like parasites. So here's a dendrobatid full of what look to be acanthocephalin worms infesting its liver. We also get eggs and volumetric data, the size of eggs, the volume of eggs, the number of eggs, and this can also provide life history data for a lot of taxa for which otherwise we actually have very little reproductive data and life history data. So Dave invited me to talk about overt, and that's what the rest of the talk is going to be about. So overt started a few years ago. This is a Thematic Collection Network grant funded by the U.S. National Science Foundation. This is $2.5 million spread across a lot of different institutions, which I'll show in a second. The goal of this was to CT scan fluid preserved vertebrate specimens. Why fluid preserved? Well, because it really is the greatest bang for your buck when you're trying to CT scan a skeleton, because all the skeleton is inside a single body. It is much, much, much more time consuming to, say, take all of the dry bones of an animal, put them in a vial, put them here, segment them out separately afterwards. It's much easier when they're all connected with each other. Our goals have been just based on what's available in US collections, and TCNs are generally restricted to US collections. Believe that we could CT scan about at least 80% of all the living genera of vertebrates. We're also setting a goal, we'll see how far we get, of providing soft tissue scans. These are contrast enhanced scans where we're actually staining specimens, mostly with iodine, and trying to do at least a family level representative across all vertebrates. And then to just put all the data up online so that they're freely available for research and education. When this was funded, it kind of surprised me, at least, by attracting a fair amount of interest initially, and that interest has really been sustained throughout the grant. You know, I don't think that TechCrunch and GeekWire are normally writing articles about grants that are basically about fish. Because let's face it, most vertebrates are fish. This is basically a project about dead fish, but yet it really captured the attention of a lot of, you know, not only in the tech community, but also outside of that in the educational communities. And I think part of that is just because of the things that you can do with these data once you have them is really appealing to a pretty broad audience. So we are now 3 years into this project. We have another year, but we'll have at least 1 year no cost extension because COVID has really delayed things. So in just year 3, this involves 120 something people. You know, we train faculty and scientists and students in the use of CT scans, both at single institutions as well as across institutions. It's generated a lot of publications. Importantly, a lot of publications by people that are otherwise not associated with overt, which is great because that's sort of the goal in the end, is to create data that's really useful for their larger research community. If you're interested, there's a little bit.ly link there that's overt_IDIGBio, where we have a full list of publications and links to other resources. We even have a YouTube channel now, which I forgot to put in the talk. So the original 18 institutions are these. You'll see a lot of familiar names. You might notice that there's 2 in here that are otherwise not known for having collections. That's the University of Chicago and Duke. Duke is involved because it supports MorphoSource, which I'll talk about more in a second. And the University of Chicago, Zeshy Lowe's lab serves as the CT scanning center associated with the Field Museum for us. Since it was funded, we have also had other projects funded. There are partner grants, so TCNs can have partner to existing networks. So we have 7 of those funded to date. So 3 that were funded in the past couple of years. One is Omega, which Omega has passed through the MVZ. The idea of Omega is to use surface scanning, not CT scanning, to scan the individual skeletal elements of vertebrates that are very large. So rhinos, hippos, cetaceans. Then we have those individual bones we have surface scans of, and you can digitally reassemble those into these giant virtual skeletons. We have a grant to mobilize data from Digimorph at UTCT in Austin. So there's no new scanning in that. It's actually just taking legacy data that's available at UTCT and making it available via MorphoSource. And that's actually already had a surprising impact. There's a lot of people that are interested in those data that are from, published a number of years ago in papers, and those data haven't been accessible. And another grant that was funded previously is Funky. Funky is led by Noe de la Sancha at Chicago State, but also involves folks at University of New Mexico and University of Michigan. And that is focused on imaging both dry and fluid preserved rodent specimens. And then we just had 4 more funded, including one associated with the MVZ. So one is CryptoVert, led by Bill Lute at Natural History Museum in LA. So that focuses on CT scanning cryptobenthic fishes. One is with Lauren Scheinberg at the CalACAD, which focuses on CT scanning and surface scanning Galapagos material that's held at the Academy, including hopefully all of their Galapagos tortoise collection. There's overt, which is led by John McCormick at Occidental College. This is an all photogrammetry-based pen. And the idea here is to provide, just like overt is providing a CT scan representing the insides of every genus of bird, overt will provide a photogrammetry model of the outside of every genus of bird. And so we would, at least for birds, have effectively an external phenotype and an internal phenotype for future work. And last, and the one associated with the MVZ, is OMISO, which is led by Emily Breaker at University of Colorado, but also involves folks at the MVZ, and that focuses on imaging Mesoamerican amphibians and reptiles. Phew. All right. Okay, so let's talk about some details of how overt works. So first of all, one of our big goals was to share data via MorphoSource. And so here's Doug Boyer at Duke, who is the lead PI on MorphoSource. When overt began, I think MorphoSource was only into maybe its 2nd or 3rd year. And I would say overt and MorphoSource are very closely related. Even though they're separate entities, we have a lot of almost daily emails back and forth talking about ways that we can improve MorphoSource, because overt is sort of like a super user of the site. So 3 years into the project, we have more than 25,000 media files for more than 8,000 specimens and more than 50 US institutions. These are all only the data that are up on MorphoSource. There's a little bit of a backlog still of stuff not yet on. These are available for the most part as stacks of X-ray images. These are zip files full of TIFF images. And then in many cases, 3D meshes representing those. So like you can see in the web browser here, this gharial skull, in MorphoSource, you can manipulate those 3-dimensional mesh files right now in the website. Most of these scans are very high resolution. It was important to us to try to get the highest resolution possible to sort of, quote, future-proof these data so that they weren't just available for a project right now, but in the future, when people actually want higher resolution data, they can actually see the sutures, they can see foramina, things like that. As much as we can generate data that's going to serve us for a long time going forward, that's what we want to do. So most of these are in the sort of 20 to 60 micron range. So these data have already had, I think, a big impact. They've clearly been used in research publications, but they've also had a large impact outside of the research community. So to date, they've been viewed more than probably about 750,000 times. This is actually a couple months old slide. Been downloaded more than 30,000 times, and predominantly for research, as reflected in publications. And there'll be more of that. It's sort of every month there's publications now coming out using these data. But a lot of the impact is also educational, and the 3D printing and art and personal and outreach. Those things also sort of overlap, where a single user is using something for multiple purposes at once. It's sort of this combination of education, art, and outreach. That use is only going to go up. So sometime soon, hopefully by next month, MorphoSource 2.0 will be online. And so you can do, I'm going to go back and then do it again. All right. You will actually be able to manipulate even photogrammetry meshes. So we can support overt in the MorphoSource site. So even color wrapped 3D meshes will be available on MorphoSource, and you can manipulate them in the web browser. But at least for overt, the thing that's going to have the biggest impact is this. So we've sort of revamped the pages, if you're familiar with MorphoSource, about the details that are available for any scan. But MorphoSource has worked with IIIF, which creates these open source viewers. What you're seeing right now is actually a view of the image stack, right? We have an ability in a web browser to actually manipulate an image stack, not only in slice data, like you might have been used previously on the Digimorph website, but also effectively have a volumetric rendering of those data. This is without segmenting those data, without opening them up in a piece of software and actually creating a 3D object out of them. We can actually still in the web browser be able to see what's inside these image stacks. I can tell you from just the impact that overt has had so far, there's a lot of people, excuse me, for instance, paleontologists that are coding characters straight off the mesh files that we have on MorphoSource. They're not downloading them, they're not manipulating them further. Having ability to, say, view the actual image stack in a web browser, I think is going to have a tremendous impact on how people use these data. Coming beyond the sort of release next month are also other tools in this web browser, such as taking just linear measurements on these objects. And I think those things, just being able to do them on a web browser without downloading them, is going to really make the impact of these digital objects kind of go through the roof. Let's talk about a little bit of the mechanics of how overt has worked. So first of all, we're kind of an odd TCN in the sense that all our stuff is, quote, already digitized, right? We're not capturing label data, right? We've already done that. So instead of focusing on an individual collection, like, oh, we're just scanning all Berkeley specimens, the goal here is not been to focus on an individual collection, but to focus on taxa. And the way that we've done this is to really build on the important work that was done by projects like FishNet and Manus and HerpNet and Ornis, because we can find where these specimens are. Not only can we find what institutions they're at, we can also prioritize ones that are, quote, better, right? If we're going to CT scan something, it'd be nice to do one that has other associated data with it, right? Not to do ones that it's from a pet store and no one knows, how old it was or where it came from or who found it or anything like that. So we use those digital data to discover specimens and prioritize specimens. So this is just an example. So it gets referred to as a hit list in overt. This is just a Google spreadsheet. And what it's doing on the left is you'll see a list of species. That is the type species of every genus. So how did we prioritize which species to do? Well, for lack of a better way, we just picked the type species for every single genus of vertebrate. So we rely on resources, taxonomic resources like reptile database, amphibian species of the world, mammal species of the world, things like that. Then what we can do with in data portals like IDIC-Bio is figure out which overt institutions have those specimens. And then importantly, which of them, highlighted in blue here, is which of those specimens we know are institutions that have associated tissue samples or images with these same specimens. Because if we're going to pick a specimen to scan, we'd prefer to do one that has, say, an associated tissue sample with it, right? Because even if there's no genetic data associated with it now, the opportunity for associating genetic data with a specimen, with a phenotype of a specific specimen, I think down the road, hopefully you can agree, would probably be powerful. In some cases, some of these genetic data already exist, but at least being able to have phenotypes for things that have the potential to get genetic data is something we wanted to set overt up for, in the long run. Okay, another thing that was really important to me, and this is as a longtime user of NCBI, was I was tired of typing in information about specimens because, and this is inspired by Jens Vindum, who many of you know. Jens was always mad about how people had typed in the data about his specimens wrong. So we wanted to figure out how to not do that. So the way it works in MorphoSource, you can type in a number. It calls using the IDIC-Bio API. It calls the information from IDIC-Bio for that specimen. You could type in just UF 12345 and it gives you the one in herpetology, vertebrate paleontology, ichthyology, and you say, oh, I meant the one in herpetology. And then we have all of that Darwin core structured metadata in IDIC-Bio. And what we do is we actually send that metadata into MorphoSource where it's stored. And this linkage really enables a lot of important things kind of behind the scenes for like reporting, MorphoSource. So obviously doing this one specimen at a time is complicated when you're CT scanning thousands of specimens. So if you have data sets like in your lab where you have, say, hundreds of data sets for a particular paper, and you'd like to make those available up in MorphoSource, there are Python scripts that are up on GitHub written by Natasha Vitek, who was then a PhD student at UF, is now a professor at SUNY Stony Brook. So that's the bit.ly link here. And you can also further customize those scripts to suit your particular needs. But the idea here was to try to have consistency among institutions and how information about like metadata, information about imaging centers, ownership, all that type of stuff was standardized across all the 18 institutions that, at least for overt, are sending specimens. And also to just make it a lot easier for people to do this, because obviously the more that you have to spend time curating all that information, looking it up in Arctos, typing it into a spreadsheet and uploading it, that's cumbersome and all is going to stand in the way of people actually sharing these data long term. So trying to develop tools to make this a lot easier was a really important part of overt. The other thing that's really great from it, and this is also inspired by Jens Vindum, is that if you know Jens spent a lot of time on NCBI just looking up Cal Academy specimens and finding GenBank numbers and then putting them in his database. Because GenBank does not necessarily curate, the collection of all the CalACAD's herpetology specimens and give it to Jens, right? So I wanted to find some way to just give it to Jens without him asking for it. So what we do, because we use the IDIGBio API, we were able to get the record set information for every specimen, right? So when institutions like MVZ publish their data, for those of you that don't know this, they're published effectively as a thing called a record set, and that's how it ends up in places like IDIGBio or GBIF. So because we had that information, it meant that we could aggregate all of the media file information and all of the usage statistics by individual collection and make it available to people, even with those that have not asked for it. So we've created this RSS feed that's up on MorphoSource. I'll show in a second. That provides a couple different files. So there's one that's a CSV file of all the Audubon core metadata about these 3D objects. At least in Florida, we just add that to our IPT. So we download it, put in our IPT, and then it gets published to IDIGBio. So that way you can actually look in GBIF and IDIGBio and see what UF specimens have CT scans. That's how it works. But the other part was actually making the usage statistics available. So there is a page on MorphoSource called Data Reporting. And for this talk, I spent a while yesterday curating information about the Museum of Vertebrate Zoology, because I thought that would be fun to see the impact that you all have had already. So right now on MVZ, and I can say, I think for the most part, we haven't done many birds, and we've certainly done no fish. So it's been mostly herps and mammals. So right now there's about 31 gigabytes of data up on MorphoSource for more than 200 specimens. And these have been picked for the most part as single representatives of different genera, to sort of capture the needs of overt in terms of having one per genus across vertebrates. Currently, those 200 specimens have been viewed more than 11,000 times on MorphoSource and downloaded more than 600 times. For the most part, these are downloaded for research, but also you see some interest there from education, personal use, and things like that. And also say, sometimes people download something and they say it's for research and personal use and art. So if you add up all those percentages, they don't all add up to 100 because people check multiple boxes when they download. On that data reporting page, you can see, the UCMP publishes their data through a separate IPT. So we have the UCMP data set here, and then we have these 3 different orange lines here, one for birds, herps, and mammals. And you'll see a media file. The media file CSV is all the Audubon core data. The downloads are all the granular download details. And the download requests are the free text of what people are actually typing in and saying they want the data for. Okay, so here's the most downloaded and used specimen from the MVZ and MorphoSource. It's this really beautiful platypus scan that we did here at UF. We have a head only and a whole body scan of a platypus. This has been viewed more than 1,100 times on MorphoSource and downloaded more than 100 times across multiple countries at this point. This is a specimen for your reference. I had to look it up. So this is a 1922 fluid preserved platypus in the MVZ mammal collections. For reference, in case you're curious how many platypi are out there in collections, there's only about 1,200 records in VertNet, and less than, as far as I can tell, less than 50 fluid preserved specimens of platypi in the US, and only about 100 skeletons. So just look at the comparison of those numbers. There's more than 1,100 views on MorphoSource and more than 100 downloads. There's actually more downloads so far than there are skeletons in the US. I feel like that's pretty amazing. Another thing that I did is I did a quick Google search for Ornithorhynchus, that's the genus name of a platypus, and MVZ to see what turned up. I only got about 25 hits, and only 2 of those actually cited a specimen in the MVZ of a platypus. What's cool is that one of those is actually a thesis recently published or recently turned in that showed up in Google Scholar. And here it is. Here is someone, Kendra Lenny, who is at, I can't remember where she is. She is, I think she's at the University of Calgary, who used this specimen in her thesis work. And while the publication, it's going to take more time for a lot of these publications to come out, a lot of times when I'm seeing Google Scholar use of data that's in MorphoSource, it's theses that are just getting turned in right now. So clearly they're having a big impact. I'd also say that we've taken that skull and put it in some collections on Sketchfab, which I'll come back to basically use for outreach and have as a tool for people teaching vertebrate anatomy and diversity. And that skull has actually been viewed 1,300 times on Sketchfab. So these single objects can have tremendous impacts beyond what they normally would have if they're sitting in the collection. Doesn't mean that we're going to scan every platypus. No, probably not. It's really time consuming to do that, but it can really expand the impact of individual collection objects. Just for fun, here's some free text of, these are only MVZ requests of people using data from MVZ. I challenge you all to find out what faculty member wanted to know about parvomolgi. But there's a lot of really, really cool projects using these data, and sometimes unexpected things, right? I mean, it's neat to see right? Remember, these are all extant taxa. There's a lot of work on ecomorphology, for instance, of extinct tetrapods shows up here. There's a lot of paleontologists using neontological data. Frog lens was one. I think that's actually a student working with Raina Bell at the Calicad. I've never thought about actually using the lenses from these scans. So it was really cool to see people even now coming up with a way to use the scans that we didn't anticipate as part of overt. The other nice part is that we capture a lot of the teaching impact of these collections as well. And so especially during COVID, there's been an enormous uptick of college faculty and sometimes K-12 educators, but mostly college faculty using CT scans to provide sort of hands-on digital objects to teach, osteology or vertebrate zoology. So there's been a big uptick in using these scans for teaching. Another important part of overt is just in general, just how broad the educational impact has been of these specimens. And I oversee a collection, so I have a sense of how many educators show up, and usually it is close to none, right? We might have someone that's really, that knows that we have a collection that maybe asks for one turtle for an art class, but we don't have a big educational impact. So these digital objects are having really a tremendous impact. So they're ending up in K-12 classrooms, they're ending up in undergrad teaching, like Chris Shield teaching comparative anatomy. They're also ending up in art classes, and art classes in multiple places around the world. So I know that there's art classes in Italy that have been using some of our data because they're teaching art students what the inside of animals look like if they're doing 3D animation or if they're doing painting and things like that. An important part of overt has been to work directly with teachers. So we've been working with mostly middle school and high school teachers to develop learning activities around these 3D data. And one of the things that this really inspired was using Sketchfab. A reason for that was we originally imagined, well, a high school teacher might want to download these data, open them up in Mesh Lab, they can do some measurements, they can actually collect some quantitative data. And we didn't quite think through the fact that, a lot of times computers are locked. They don't have admin access. They can't install something, even if it's a free piece of software like MeshLab. A lot of times people are teaching on Chromebooks where you can't actually install any other bit of, sorry, I have to answer my phone. That was weird. A lot of times you have people teaching off of Chromebooks, so they can't actually download and install things on their computers. So one of the advantages of using Sketchfab is it's all web-based. And so we've put pretty much all of our digital outreach through Sketchfab right now. And one of the reasons for this is that we can do things like this. So here's a nice Dermophis Sicilian skull. You'll notice it has points on it. A thing that we can do in Sketchfab we can't do elsewhere is we can label them and we can provide annotations, and these have proved very useful for teaching. We can also call by a URL images that are on Flickr or Wikimedia and embed them in the annotations, right? Because not everybody knows what a tentacular foramen is, so being able to show a tentacle of a caecilian there is actually useful. We've been further doing things like this. This is led by Jamie Gray, who's a postdoc with us, who's based in Australia right now, of actually segmenting out limbs. And so this was inspired by a teacher who wanted to be able to teach about homology and diversity. Segmenting out limbs, color coding them all similarly, and then being able to reassemble, all of vertebrate limb diversity into a single view, so that with a single URL, a teacher could actually talk about, say, the fin to limb transition and actually have, similarly positioned, similarly colored forelimbs to teach teach ideas about homology, diversity, convergence, and more. So I'm gonna have a few more slides to hopefully wrap up. So, what does the future hold for this? So one of the inspirations I've really had is from Notes from Nature. Probably many of you know what Notes from Nature is. It's basically a platform that we can use to bring in collections data like these botanical sheets or these slides, and actually interact with community scientists or even students and colleagues like ourselves around the world that can actually annotate and provide new data associated with these specimens. Sometimes it's data that's otherwise not present on these sheets. So for instance, you might have a project that asks for flower. Does it have a flower, yes or no? Because in the digital record, it says nothing about a flower, right? So this was sort of put forward by some postdocs in the NSF PRFB program as digitization 1.0, right? The initial part was just simply creating all these digital objects that represented things in our collections. And the second version of digitization was creating these workflows so that anybody could add new layers of information to those digital objects. And then further, that it didn't really matter where in the world they were, right? So that they could all be around the world and collaborate on the same digital object, where otherwise, if we did this in real life, we'd have to mail that single specimen to all of the people working on it to take different traits, right, separately, which is very cumbersome and time consuming. So being able to have these digital representation means that many of us at once can collaborate and work on something. So here's my little science aside. So during COVID, we were looking for lab projects because suddenly we could not be in the lab or the museum anymore. So we brainstormed. We had sort of this project had been kind of percolating in the lab for the last year. And then a student in the lab kind of took it off because they really needed the project to work on because they couldn't actually be in the lab doing anything else. So here's a simple one. Here's an example of a research project that has really only used pre-existing digital specimens, and it is tooth loss. So the loss of teeth is actually relatively rare in terms of number of evolutionary transitions across vertebrates. We have a single loss of complete loss of teeth in fishes. That's signathid. So most other fish, if they've lost them in their jaws, they still have them in their pharyngeal jaws. So we have signathids like pipefish and seahorses have lost teeth. There's no example of squamates that have lost teeth. Turtles, or at least as far as we know, represent one single loss of teeth in their evolutionary history. Same at least with crown group birds, although there's some dinosaurs and things that have also independently lost teeth. And there's 3 extant mammals that are completely toothless. There's other mammals that lack enamel and things, but there's 3 that are totally toothless. And looking across vertebrate diversity, this has actually provided a really cool system for understanding how the repeated loss of a trait is related to this sort of parallel degradation of these molecular genetic networks that, that cause this trait. So there's been a lot of interesting work done, especially in the context of mammals, about tooth loss and enamel loss, taking advantage of the fact that multiple groups have convergently lost teeth and looking at, say, the enamel genes and how those have degraded independently across these lineages. You'll notice there's no amphibians on here. And actually one of the most sort of interesting cited thorough reviews is this one here that was published a couple years ago, and actually only talks about toads, which is kind of remarkable if you're an amphibian biologist, because I feel like any amphibian biologist can start listing off all the frogs that don't have teeth. So we thought, well, we know a lot don't have teeth, but there's actually never been really a synthetic study of this before. So all frogs except one lack teeth on their lower jaw. Some frogs have them on some frogs lack them on their maxilla and premax. And then the absence of teeth on the vomer varies quite a lot. So this is a project led by Dan Palou in the lab, who was casting about for a project since he couldn't be in the lab, but also involves a lot of other people in the lab, including people who were not even in the same state or country during this time, who are collaboratively helping collect these data. So this took advantage of the fact that up on MorphoSource, we have about 1,000 frogs right now, representing all species and almost every single genus of living amphibian. So what we do is we'd open up a page like this. We would look at the mesh file. Here's this nice frog. We'd rotate it, say, oh, there's teeth on the maxilla, teeth on the premaxilla, teeth on the vomers. Just double check there's no teeth on the dentary. Nope. Okay. And then we would record those data in a spreadsheet. It was as simple as this, really, as a lab-based project. And the nice part here, too, is that we're immediately able to associate traits to media files, which are associated to specimens, which are associated to taxa, right? So we have this sort of chain of these connections. I like that Becca is smiling right now because she's heard me talk about doing something like this in Amphibian Web. So this is exactly like how we've been doing things in the lab. So what's really cool, this is a project that we did based on data we already had. And amazingly, when put in a phylogenetic context, this suggests that amphibians, and in this case, only frogs, because salamanders and caecilians all have teeth. There's been 19 independent losses of all teeth in frogs, which is remarkable. There's been a lot written about how important mammals are as a system for studying tooth loss, and there's actually only 3 complete losses of mammals' teeth. We have a lot of opportunities here for taking this as a system for then actually asking questions in a sort of phylogenetically structured way about tooth development, about tooth genetics. And that's actually sort of the next step of things we're doing in the lab right now. So that was my one little bit of science. So, but imagine being able to actually do this in a Notes from Nature platform. Here's that same skull. We have a little project where maybe we can use the MorphoSource API to call in all the mesh files of frog skulls. They appear here. We have community scientists on other sides of the world say, oh yeah, teeth are present, teeth are present, teeth are present. They record those data. That's powerful. It enables us to actually get data for vertebrates that are phenotypic data that otherwise are not associated with a record, right? No one said that this frog has teeth in its specimen record. It enables also a large audience of people to help do this. So, if we only have 100 or even 500 things to do, 500 things my lab could handle on its own with 8 people working to do this. But, we have 15,000, I think, core dates right now in MorphoSource. If you wanted to do a project that synthesized data across all those, that would be very challenging. So one approach would be to use, community scientists to, in a format like this to interact with those data. Another would be to basically use what we've done already as effectively training data for using deep learning methods, forms of artificial intelligence, to use those training data to then actually train on all the data we don't have any information for, to help us find the answers through all those thousands of data sets that are very time consuming to open one at a time on your computer. So just some thought-provoking things. And this is actually something that we're in the midst of trying to write a little proposal related to. So they're kind of on my mind. So we have labeled data sets. So that's something that you need in AI, right? You need say, hey, this is a picture of a bird. Like the bird is actually a label, right? So we have data sets that already have taxon names. We already know what all the snakes are. So given that we have all these image stacks where we know all of those that are snakes and all of those are frogs, can we actually do an intersect and say, I want to know all the snake data sets that have frogs? Because it turns out frogs turn up in snakes, right? Because they eat them. We can probably use some probabilistic clustering methods as well on these same labeled data sets to discover morphological groups, to detect outliers, right? We were scanning thousands of fishes. Clearly, there's a lot of fishes that people already care about and know about, but we have taxa that nobody's ever turned their attention to. So without opening every single one of those thousands of scans, how do we know that something's already interesting there, right? So being able to train, a deep learning approach to find outliers could be a really interesting approach. And last, if we already have image stacks from which specific bones have been isolated in mesh files, like we just published a paper on the humerus of a lot of frogs. Can we use that as a training set for isolating the same bone, from other data sets without having to necessarily do all of that segmentation work? And these are things that we're just now turning our attention to. And I'll say that, a lot of these things have not really been that important to do when we had 10 data sets or 100 data sets, because realistically we could have an undergrad help and get it done. But now that we have tens of thousands, that is a lot more intractable. So we really need help from potentially some deep learning approaches. All right, I'm wrapping up. So I've talked a lot about digitization and accessibility. So there was a paper published a couple years ago. Actually, I think this is wrong. I don't think this was 2020. It says 2020 on here. I think it was 2017. So this is published by Josh Drew and Corey Moreau and Mel Stiasny. That was about this issue of whether or not digitization had the ability to increase accessibility. And and a lot of times in our collections, when we talk about accessibility, we tend to mean like more researchers like us can access those data. We tend not to mean that a more diverse audience of people that otherwise normally wouldn't interact with these data. So how do we really think about we have these accessible collections. How do we ensure that they're actually reaching a more diverse community of users? So, for instance, in the art and cultural heritage world, there has been a focus over the last 20 years on decolonizing museums, of recognizing the fact that sometimes the objects in our museums were basically obtained because of different features of colonialism, that there's racism embedded in our collections. A lot of times in the art and cultural heritage world, decolonizing museums really focuses on the impact you can have in the public floor, right? It talks about surfacing these stories of hidden contributors that had big impacts that otherwise are not well-known scientists, talking about those figures to the public. But what does that mean for the MBZ? It doesn't have a public floor. How do you begin to think about taking a collection at a university museum that doesn't necessarily have that public impact and still trying to find ways to diversify the audience that interacts with these specimens, right? So, there's some obvious approaches. One might be to support the training of scientists and students from the countries where these specimens are coming. You have a really important collection of specimens from Mexico. Maybe your institution should double down on really working with Mexican scientists, right? So a lot of the digitization work we've done has led me to think about these issues. And if you're interested in these issues, I'd encourage you to join us with the Education Outreach, Diversity, and Inclusion group within IDIG Bio. There's a small group of us. We currently meet once a month. We meet a couple times a month, but we have open office hours once a month, at the first Thursday of the month at 11 a.m. I appreciate that's a little early Berkeley time. If you're also interested, right now we have a SACNAS booth. So the SACNAS conference is happening right now. So we have a virtual booth at SACNAS. You can come chat with us. And the last thing I'd say, we're really interested in growing the diversity of users that interact with our collections, so we can make this more diverse community of museum scientists. So one of the things at IDIG Bio we've been doing is actually developing a program that targets high school students and college students to introduce them to biodiversity and museum collections. That's something called IDIG Trio, which is a partnership with the Department of Education supported Trio programs here at UF. And I just encourage you, if you have students, send them our way. I mean, we're basically going to do virtual shadowing, virtual career fairs and things like that this coming February. Last, I would be remiss if I didn't thank all of these people and more who are not on this slide. That's basically who powers everything that we do in the lab. And I'm happy to spend some time talking about frogs or CT scanning. Thank you.
Yay.
Well, thank you. I'm glad somebody clapped.
I'll clap also.
I found that to be the most awkward part of Zoom talks at this point.
I think we need to have a change where it's expected for everyone to turn on their mics and clap.
Exactly.
I had to include my own laugh track in my recent seminar for the department just because it was really sad to tell jokes and not have anybody laugh at my jokes.
That's pathetic, Dave.
Here I am. There you go. So, questions?
Yeah, so let me just, I'll mention that people can ask their questions verbally if they'd like. People can use the raise hand tool there to simplify things if there are a lot of questions. And if anybody wants to ask questions via the chat, I'd be happy to read those for you if you're bashful about verbally asking a question.
I have a question that I can jump out there. So Dave, I wanted to ask a great talk. Yay. Thank you for talking about overt with everybody. And I wanted to ask how many people that had CT scans from a long time ago do you think have been able to get their stuff on MorphoSource through this project?
That is a great question. I don't have specific, well, this project is not overt in that case. This project would be MorphoSource. So
Right.
I don't have a good, clear answer on that. I can say that it's a conversation that seems to happen like at every conference I'm at, to say, you know, because people, it's time consuming, right? It's been time consuming. There hasn't been a good way in the past. A lot of times, at least if you care about collections, the ways that have been out there in the past, like, oh, it's in a Dryad package or it's up on the Open Science Framework. That stuff is so divorced from scientific collections. If you care about, you know, where those objects are from the collection point of view, they're really hard to find. I think people have been doing it. I have a lot of conversations with folks where they say, oh, I had an undergrad like 5 years ago that did a bunch of CT scanning and we didn't do anything with it. You know, what should I do with it? I said, put it on MorphoSource. Send it to us and we'll put it on MorphoSource. So that happens pretty frequently. Hopefully more. Actually, I can say and hopefully any of you that are reviewing papers where CT data might appear, you have the same reaction of these data should be available. And MorphoSource is a good option because it actually, you know, enables collections to know that their, you know, scans of their objects are out there.
Any other questions?
Yeah, so I see your question in the chat. How many non-USA specimens are in MorphoSource? There's quite a few. It's a little tricky because you might notice, if you really look hard in MorphoSource, you'll realize that there are specimens in MorphoSource that do not appear on the data reporting page. And that is because sometimes they're from collections that actually don't otherwise publish their data, right? They have no IPT, there is no record set. So we're not going to aggregate data from a collection, a small collection, or even a big collection that otherwise has no digital footprint. I can say from my own lab, there's a lot of those data that are up there. From overt's point of view, you know, non-US collections are supposed to officially be out of scope. Although there's definitely some non-US collections that have cropped up here and there. But you know, that is happening. Sometimes it takes some sleuthing. The second version of MorphoSource 2.0, which I see your question, Gwen, any day. It's kind of been any day for a long while now, but hopefully by the end of this month, the targeted date I think was, what is today, the 21st? I think the targeted day was actually like a week from now. So hopefully by the beginning of November, that'll be up there. And one of the advantages of that is actually having a little bit better organization of the data from the collection point of view. So they've spent a lot of time. One of the big delays has actually been curating all of the acronyms for the abbreviations like MVZ or UF for hundreds and hundreds of institutions, some of which turn out to be personal collections. And so the reason they're doing that is to actually have effectively like pages for every institution. So it's easier to actually find and organize data from different collections.
So Dave, I see that Becca has her hand up, and then maybe after Becca's question, you can address Carla's question that's in the chat.
Yeah, I was, so I hadn't heard of the Nature Notes website that you mentioned. That was pretty cool. Does it allow, you know, can multiple people provide an answer for the same specimen so you have some kind of quality control?
Yep. And so a really cool thing there is to have, you know, have the same object. So a lot of times it's Notes from Nature, which is supported by the larger platform Zooniverse. A lot of time that's been really important for transcription. And, you know, this is the vertebrate crowd, right? So vertebrate crowds tend to not use it. So it's Notes from Nature has been used a lot in botanical collections, right? Of scrawled handwriting on botanical sheets where people are trying to read it and they can actually have the same thing transcribed 3 different times, right? So you have that. But it's also been used a lot in phenotyping. So people that are actually interested in knowing, you know, what phenophase is it? Does it have flowers? Does it have buds? And to get that data, you know, at least 5 times independently.
Mm-hmm.
Yep. There have not been a lot of, as far as I've seen, real animal use cases other than like transcribing a handwritten ledger, for instance. But I think there is an avenue forward of, you know, trying to engage the community that studies vertebrates with 2D images. And certainly it's not there yet for the 3D data, but something that, you know, hopefully we can kind of push forward to with the MorphoSource API in the next couple of years.
Very cool.
So would you like to jump to Carla's question? Do you see it there, Dave?
Yeah, MorphoSource, Carla, has been supported in part by Duke and also supported by NSF. I know this is something that Doug Boyer thinks about an awful lot. And I think I'll leave it there. The best person to talk to and ask those questions to is Doug. But Doug has actually been really working on sustainability plans for MorphoSource, because clearly it's important, right? And I'll also say, you know, a reason that we chose MorphoSource rather than put things into collections or put things in IDIG Bio was, right, like collections are not set up, like Specify and Arctos, they're not really set up to serve, you know, gigabyte files per specimen, right? That's cumbersome. And, you know, a lot of the metadata that associates with those scans, we don't really have in our collection databases right now. So that was one. The other was that, you know, as part of IDIG Bio, things related to TCNs, it's supposed to appear in IDIG Bio. But again, IDIG Bio can't take these CT scans. So, you know, where are we going to put them? And I'll just add that I know Michelle, if you guys have questions, I'm going to point you to Michelle. Michelle was at a workshop earlier this year that Doug and I ran at Duke that related a little bit to some of these ideas. And there are institutions, some of you know Nelson Rios at Yale, where, you know, Nelson actually has basically a framework where he, you know, is able to know what's on MorphoSource. They download those data. They actually put those in the Yale archive. So they locally at Yale also have a copy of those data, and they add those to their database so that you can find it, you know, in their database that they actually have CT scans available. And different institutions have done really different things because, as you would know, institutions are all different from each other. And so, you know, sometimes people just do not have a Nelson Rios to actually, or a Michelle Koo to figure out, you know, how to do those things.
So I think the next question on the sequence here, Dave, is Aaron Westein's collection about file formatting.
Yeah. So this new slice viewer, yeah. So it's a sleight of hand, and I'm going to do my best to explain it, and I don't fully understand it. So my impression is what's happening here for TIFF stacks, and I think, yes, they can do this for DICOM as well. You know, a TIFF image and a DICOM, those are big images, right? And serving, you know, a stack of hundreds of those in a viewer, it's going to computationally for your computer going to be expensive. So what they actually do is they've created for all of these data sets effectively a low res JPEG based version of that data set. And it's with that lower res version that it's actually easier then to manipulate in the web browser. And I believe that that will be true whether or not it's a DICOM view, or like a DICOM file or a TIFF file. Yeah.
Can I ask a follow-up, Jim?
Sure.
I wonder what you think the sort of utility of that is going to be. Like, do you think for people who have a lot of data coming hot off the scanner and they don't have the time to like create the meshes or something, people can actually use that viewer? And I think you mentioned, but I don't really remember, can they do measurements right from that view and landmarks?
So that's two different ways. So the first about measurements. So I actually am not completely confident that the version that's going to appear in the new MorphoSource right now is going to allow measurements. However, I do know in the demo tool, it does allow measurements. And I think they're still kind of fine tuning that because there's some kind of quirks to it. You can't yet store them. One of my things I've been advocating for is actually, if you take a measurement, store it, say head length, link to the anatomy ontologies and say head length and store it so that you could then come back to MorphoSource and say, I only want to see heads bigger than 20 millimeters, right? Because if somebody captures that data, why are we just throwing it away? But in the short term, hopefully the viewer would be able to have at least a node-to-node measurement tool. And that takes advantage of the fact that we know, associated with each file, what the voxel size is, right? Because otherwise it's just a line, so we know how long the line is.
Well, that's why I was wondering about the DICOMs versus TIFFs, right? Because
there it's embedded.
DICOMs have that. Yeah.
And then your other question was about what? I forgot.
Oh, I was just trying to think about, you know, how this is going to sort of increase usability, because I know for me, at least, I get a bunch of TIFF stacks back, and then it maybe takes me months or however long to, you know, render them and then throw them up on MorphoSource. So okay, great question. So you might ask, why are there not images or mesh files of everything from overt? And that's because NSF counted that as research, and they don't fund research on TCNs. So the meshes that are there technically from overt are outreach. So there's not 3D meshes for everything. So that's part one. The other part is, I'd say MorphoSource has already been used in really weird and surprising ways to me. So for instance, my inclination is to think, well, if you found the data set, you would download it, you would then open it in one of these pieces of software we use, you'd make a model, you'd take a picture of this nice model. People are screenshotting straight off of the web browser and putting it in papers.
That's happening pretty regularly. Doug and I recently had a back and forth with Peer J about what the copyright was. They're like, well, you can't copyright a CT scan, so you can't copyright a screenshot of a picture of a CT scan that you took yourself. Like, I don't even think we need to worry about it. It struck me as one of those things where it's like, I can't believe someone's putting this not great image in a paper. But for people that otherwise don't have access to expensive software or the computers to do this work, it makes sense. And, you know, to some extent, it conveys what they're trying to convey. I think it's going to be interesting to see what happens when you have the volumetric viewer. I promise you there are going to be people that code characters straight off of the volumetric data without ever seeing a mesh file. Is that a good idea? Probably not. But at the same time, it's going to happen.
Okay, thanks.
Your next question is Jack's at the bottom of the chat.
Yeah, this is a good question of who is best in the position to curate and handle these digital collections. Well, I feel like it's a good question for the MVZ staff of, you know, it's time consuming to have to also, you know, curate these objects. It's just adding on to work that otherwise you don't have time to do, right? Because all collection staff is usually already spread pretty thin across a lot of tasks. So one thing that's been incredibly cumbersome in MorphoSource has been approving downloads. And why is it set up like that? Well, one of the reasons was because there's long been kind of uncertainty about copyright. Was there copyright that you should apply or not? I think pretty much we've all come to the agreement you cannot copyright a CT scan. There's actually a legal case about that for human data, but even for museum specimens, probably not. You can't copyright it. Likewise, you probably can't copyright an actual image of a specimen because it's supposed to be a, quote, slavish reproduction of a real thing, which thus is not copyrightable. Nonetheless, museums are interested in controlling access to these data, right? There is actually a commercial interest in them. If it's a paleontological specimen, there's certainly a commercial interest in them. So one of the reasons that we've had, you know, all these data behind, you know, requests to download has just been to try to limit that commercial use because we really had no other mechanism to enforce. And I approve download requests all day. That's like one background thing I'm always doing while checking email. But hopefully that will stop. And the reason for it is because I think we have a better framework within MorphoSource for user agreements. It's instead of, you know, governing access and control of these data via copyright law, it should be via contract law. And so really trying to develop a contract and a usage agreement with users when they download things about what they're going to use. So if you really wanted to try to go after somebody for, you know, using something and, you know, making a lot of money off it, like Pixar, for instance, you would have actually a legal challenge to do so. And you might laugh and think, why would Pixar want CT data? Turns out they animate a lot of animals. And we actually get a lot of requests from artists that are really interested in using, you know, crocodile skulls because they are trying to animate, you know, a crocodile skull for a big movie. And that actually happens fairly regularly at this point. Not to say that you shouldn't allow them to do that. And maybe you want to do that and just give it to them for free. I think that's fine. There's certainly institutions that are interested in having a conversation at least about, well, you know, could some money come back to the collection, you know, in order to support grad students? And I think that's, people feel philosophically different about that, but I feel like it's at least a conversation worth having, especially for collections that otherwise don't have resources to support curation, right? If they can get some money off of a digital object for Pixar, and you can send a bunch of grad students to, you know, go to a conference or help with curation, I feel like it's a conversation worth having. So.
I think we'll just have one more question. The last one on the chat here from Juan Liu.
Oh yeah, ooh, this is a good one. Yeah, what happens when you have so I don't know is the answer. I'm interested in this. We've been talking about what to do with this. We've talked about this within overt because clearly, you know, a great thing would be having CT scans of model organisms because clearly there's a big community that already cares about that organism. Technically, we're only supposed to scan things that are already in collections. And so, you know, do you curate that collection of model organisms and put them in a collection and then scan them? I don't know is the answer. I think Doug would be a person, Doug Boyer at Duke would be a person to ask that question to. I think it's really important because I think there's probably a lot of really interesting phenotypic data that's disappearing because it's not museum based. Maybe it's up on the Open Science Network or something like that. But clearly there's the availability of these machines on campuses means that more and more people are actually using them for, diverse research and, even model organism.
So Dave, can I follow up on that? I mean, are there researchers doing like Evo-Devo work that use CT scanning as part of their visualization? And then what happens to all that material? Where's it going?
I don't know. But an interesting question is, more broadly even than CT scanning for Evo-Devo, is what happens, sorry, I'm changing the subject entirely. What happens to Evo-Devo material? Just like 20 years ago, as you had collections where, the tissue collection was the lab freezer and then people retired, those tissue, now we have tissue collections in museums. What about all of these other materials that are associated with Evo-Devo labs? What will happen with that stuff? It would be a real shame if all of those materials that are in freezers that can be used for in situs or antibody studies are just tossed. Should museums think a little bit more about cultivating a relationship with Evo-Devo? And that would also relate, to thinking about their digital data as well, right? Because there's definitely labs in Evo-Devo creating a lot of digital data.
I think they'd swamp us pretty quickly.
Hey, they could, but they also write NIH grants, man. It's not a bad thing for a museum. So I feel like it's an interesting conversation. They're more like us than, model system people, right? They care about a lot of the same topics that we might care about. Okay.
That's great. Why don't we all unmute and thank Dave for a really amazing talk, and very much enjoyed the question and answer session afterwards as well.
Thank you, Dave.
And I'll just say, if people have further questions, happy to, you all know where to find me. I pretty much live in Zoom. So I'm here if you want to talk.
Thanks, Dave.
Thanks so
Thanks
much.
so much.
It's great to
Thanks
see
very
everybody, by
much.
the way. Really nice to see all of you.
Thanks,
Terrific,
Dave.
Dave.
Loved it.
Thank you.
Good to see you. Thank you.
Dave, yay, MEZ shirt.
Good luck tomorrow, Dave.
Thank you.